Hey, my name is Johan Skog. I'm the Chief Scientific Officer at Exosome Diagnostics. And today I'm going to talk a little bit about the current state of exosome-based biomarkers. And this presentation will have two parts, where the first part is an introduction to exosomes and other microvesicles. And then part two is a summary of the EPI test, which is the world's first exosome-based biomarker test for prostate cancer. So to set the stage a little bit and give you an overview of what exosomes and liquid biopsy really means. When people are talking about liquid biopsies, we often mean detection of molecular markers such as RNA and DNA without the need for a tissue biopsy. So you can basically surveil and monitor molecular events without the need for an invasive procedure. So this is applicable to a wide variety of diseases, of course, because this is minimally invasive. It overcomes some of the heterogeneity of tissue sampling. And you have liquid biopsy analytes released from a variety of processes in the body, not only the tumor, but also tumor stroma and immune cells, etc. So it can give you a broader picture of what's going on inside the body. When we talk about exosomes, these are small vesicles that are actively released from cells into the surroundings. So these exosomes end up in the biofluids. So you can isolate exosomes from blood, from both plasma or serum, from urine samples, cerebrospinal fluid, saliva, etc. However, the source of the exosomes varies depending on what biofluid you are looking at. So this is a schematic image of tumor cells growing in the body. And you can see this cloud of vesicles being released from the tumor cells. And we know that a cancer cell can release over 20,000 of these vesicles in a day to the surrounding. And what we've learned is that these exosomes are packaging the content from the donor cell that released it. So you can find all the RNA species, proteins, and lipids that are coming from the donor cell inside these vesicles. So it basically gives you an opportunity to find what's going on inside the tumor cell in real time. So exosomes are released by all living cells into biofluids. However, cancer cells release many more of these exosomes. So they're produced and they play a role in health and disease. However, cancer cells utilize exosomes to its own advantage. So we know that cancer cells release exosomes that stimulate tumor cell growth. And cancer exosomes can also inhibit immune cells, so they can block the immune response on both the adaptive and the innate immunity level. So tumor exosomes block NK cells, macrophages, as well as T cells. They can also stimulate angiogenesis. So they are playing a role in all of the hallmarks of cancer formation. We also know that exosomes package material such as RNA and proteins that can be used for intercellular communication. So these vesicles are actually a new form of communication that wasn't appreciated before, where historically intercellular communication happened primarily through secreted proteins like hormones and peptides. However, with the exosome components, you can have intercellular communication with molecules that are not normally secreted. The key aspect of utilizing exosomes for diagnostic purposes is how you get to the exosomes, how you isolate them. And here at Exosome Diagnostics, we've been optimizing that process for over a decade, where we have optimized cGMP manufactured isolation platforms for exosome isolation from various processes. So we optimized it for various biofluids, and the method of choice depends on the biofluid and what you're trying to do with the sample after. So one example is the Exolution UPrep here, that is the optimized kit for urine exosome isolation. Exolution EDDI is an example where if there is a need to isolate tissue-specific exosomes or disease-specific exosomes, you can enrich your sample by immunoprecipitation, where you either enrich or deplete non-relevant exosomes from your sample, and you can therefore increase your disease-specific signature. If you do your isolation properly, you can do a complete RNA profiling of the exosome, and you can see the entire RNA transcriptome in exosomes. And you can do that from any biofluid. So we've optimized the process from plasma, from serum, from urine, and even cerebrospinal fluid. And we can work with as little as 0.5 ml of plasma or up to 10 mL of urine. And if you do your sample preparation correctly, you can actually get the same diversity of RNA and the same amount of RNA as from tissue. So basically, looking at 0.5 ml of plasma, you can see and interrogate over 30,000 of the 56,000 genes. You can interrogate over 17,000 out of the 20,000 mRNAs that are known. And these are similar numbers as you get from sequencing a tissue sample at this read depth. It's a highly sensitive way and to look at different RNA molecules in circulation. For some applications, you do want to have an enrichment or depletion procedure in place, because if you're looking in a plasma sample, for example, you have over 10 to the 11 exosomes in circulation in 1 milliliter of plasma. So these are extremely abundant events. However, only a fraction of the circulating exosomes may be coming from the diseased tissue. Especially if the tumor is very small or localized to a certain region. And in this case, if you're looking at a marker that is not unique to the disease, you may want to do an enrichment or a depletion step. And you can do that through surface biomarkers or surface markers on the exosomes that are similar to the cell that released them. So this EDDI platform, it's an abbreviation of Exosome Diagnostics Depletion and Enrichment Platform. That enables you to have a window into what happens into a disease or tissue-specific organ. So you can look into the brain, for example. You can get brain-specific signatures out of plasma, which is a game changer. You can also look at earlier cancer detection and RNA profiles that are similar to tissue. In some cases, your signal is hidden in the noise when you're looking at a total plasma sample. One example of that is, for example, alpha-synuclein, which is a protein that has a lot of interest as a biomarker in neurodegenerative diseases such as Parkinson's disease. We cannot, if we're looking at alpha-synuclein in plasma, it's extremely high because alpha-synuclein is also produced by red blood cells, and that's coming from a disease-irrelevant source. So it has a lot of background signal that is blurring the noise. However, if you do neuronal exosome enrichment from the plasma, you can actually get the signal out of that noise. And it enables you to do disease-specific protein analysis as well as disease-specific RNA analysis. So this has applications in a variety of diseases, and here's an example where we applied this in early cancer detection signatures of breast cancer patients. So these are women with ER-positive, HER2-negative cancer, and as controls, we had women with nodules that were confirmed to not be cancers. And we had plasma, 1 milliliter of plasma, and a matched FFPE tissue from the actual nodule, from the tumor, or the benign process. And when you're doing RNA sequencing and you're looking at the RNA transcriptome, the benign tissue is, of course, different than the cancer tissue. And when we're looking at the total plasma exosome, we also see that the cancer individuals are different than the ones that did not have cancer. However, the RNA signature is not the same. So even if you can differentiate the patients from the controls in the plasma sample, they are not identical to what you would see in the tissue sample, unless you do positive selection of breast cancer-specific exosomes. So in this example, from the same individuals, we did positive selection of breast cancer-specific exosomes. And to our surprise, we saw a very nice correlation between the signature in blood and the tissue. So when we looked at the RNA targets, you can see that the plasma matched up and clustered next to its matched tissue. So the blood sample, from the blood sample, not only could we tell you that you were a patient or a control, we could also tell you from what blood sample, what tissue sample the blood sample corresponded to, which is a really unique application of these exosome enrichment strategies. So as you can imagine, this opens up a lot of different applications that you can utilize the exosomes for. So over the last decade or so, here at Exosome Diagnostics, we've been working on a lot of different types of tests that are looking at RNA expression profiling in biofluids for monitoring of treatment response in clinical trials. We have the EPI test, the prostate cancer test that is commercially available, and we'll talk about that in part 2 of this presentation. We also have mutation detection assays as well as other platforms. So we like to say that exosomes can be used for anything on the planet and beyond, because we know that the sky is not the limit for exosomes, because we can utilize exosomal platforms also for applications outside of Earth. And here is an example where we worked with NASA for the last several years, showing that we can get profiles from a neurological syndrome that affects astronauts in microgravity. When astronauts go up in space, they get this VIP or SUN syndrome that leads to higher intracranial pressure and visual impairment and things that you don't want to have when you're flying in space and trying to land a space shuttle somewhere. So it's important to understand what's going on inside the brains of these astronauts. And since tissue sampling is not an option here, looking at the brain-derived signatures in the exosomes, it's actually a very desirable platform. And that's the end of part 1 of the presentation. For part 2, we're going to talk about the EPI test, which is the world's first exosome-based diagnostic test. It's a urine-based liquid biopsy that utilizes a 3-gene RNA signature for risk stratification of high-grade prostate cancer. So as I mentioned in part 1 of the presentation, exosomes are a very rich medium for biomarkers, and we can isolate all of the RNA transcriptome and monitor over 30,000 different RNA targets in these samples. However, for the EPI test, we are only looking at 3 of those genes. So in the prostate cancer space, we know that there's an issue with overdiagnosis and overtreatment. In the US, there are about 1.2 million US prostate biopsies, and unfortunately, not all of them are finding treatment-worthy cancers. So there's a lot of benign and low-grade cancers identified with these biopsies due to the fact that PSA has a fairly low specificity as a rule-in test for biopsy. So new biomarkers should be designed to risk stratify the high-grade prostate cancers, and we define them as any Gleason 7 and above. So Gleason 7, 8, and 9. And the intended use for the test is basically for any man that walks in with an elevated PSA in the 2 to 10 range, which we consider the PSA gray zone, where PSA does not perform very well. And it's for men that are 50 years and older presenting for initial biopsy. The workflow of the test is very simple. The patient just have to urinate in a cup, and it's a first catch urine sample that's very important. So the first 15 to 20 milliliters of urine that is sequestered into the cup, and that is sent into Exosome Diagnostics for analysis. It's important to note that the urine sample is a standard urine sample. In other ways, you don't want to have, or you don't need a DRE prior to the urination, and it can happen anytime during the day. The only restriction is we would like to have a urine sample within, or the patient could not have urinated within 60 minutes or 1 hour before the sample has been collected, because that is the amount of time it takes to regenerate the prostate exosomes into the urethra. So once the sample arrives at Exosome Diagnostics, we isolate the exosomes, we crack them open and get the RNA out, and then we do a qPCR analysis of 3 genes. These genes are PCA3, SBDF, and ERG. The result, the quantitative analysis of these 3 genes, then goes into an algorithm that converts this into a single score. And this score typically falls in on the range of 0 to 100. And if you're below 15.6, you're negative. So remember, this is a test that is developed to avoid unnecessary biopsy. So it's a rule-out test that is optimized for its high negative predictive value. The performance of the test has been extensively validated. So the first prospective multi-institutional trial that we did, that was published in JAMA Oncology in 2016, achieved a sensitivity of 92% with a negative predictive value of 91%. So remember, the negative predictive value is what the test was optimized for, because it wasn't optimized to rule patients in, or the cut point is not optimized to rule patients in. The cut point of 15.6 is to rule patients out from unnecessary biopsies. If you look at the negative predictive value of 91%, that includes in this study 12 patients that actually had a Gleason 7 that were missed. So of those 12 patients, 9 of them were Gleason 3+4 with less than 3 positive cores. So if you define the high-grade disease as Gleason 4+3 or higher, the negative predictive value goes up to 97%. We actually did a second independent prospective validation trial as well. And if you combine those two independent prospective validation trials, you see the result here. So basically the two studies showed near perfect performance overlap between the first and the second validation study. So the overall sensitivity of the test, when you combine over 1,000 patients here, has a sensitivity of 93% with a negative predictive value of 90%. And if you define high-grade disease as Gleason 4+3, the NPV goes up to 96%. We also know that even though we have a well-validated cut point of 15.6, we know that the EPI score correlates with high-grade prostate cancer in a way that the higher the score, the higher the chance of finding high-grade prostate cancer upon biopsy. So having a cut point here around 15%, you can see, or EPI of 15.6, as this EPI score goes higher, the higher your likelihood of finding aggressive prostate cancer, so grade group 2 and above. You receive a report back that is showing you the scale from 0 to 100, and anything below the cut point here is shown in green, and you get the actual score for the patient where it lies on the scale of 0 to 100. And today we have a median turnaround time from sample arrival in the lab to report is about 2.1 days. However, we guarantee a turnaround time of less than 5 days. So to summarize the EPI test, it's a simple, non-invasive, non-DRE urine-based test. Importantly, this is an algorithm that is independent of PSA or other standard of care factors. So the algorithm for the EPI test is not influenced by the PSA level or family history or other risk factors. So we can use EPI in conjunction with those standard of care risk factors in the assessment for the patient. And this is an important discriminator for the EPI test. Because if you do have an algorithm that weighs those factors into your score, those risk factors are already considered in the test and can't use it twice in the nomogram. So the EPI test is validated in 2 prospective U.S. multi-institutional studies that includes academic centers and lipo groups. The first validation study was published in JAMA Oncology in 2016, and the second validation study that had near identical results was published in European Urology in 2018. So the test is designed to reduce prostate cancer overdiagnosis and overtreatment, so it's optimized for its negative predictive value. However, as you saw on the graph on the previous slide, there is a increased probability of finding cancer the higher the score. EPI is now included in the NCCN prostate cancer early detection guidelines, and FDA has granted breakthrough device designation to EPI.